One of the things that you'll find in the description on the module, and also when you look at these diagrams, is that you'll find that the cortex has a granular appearance. You see these like dots? Has a granular appearance, and the medulla has a striped appearance. So what is it that gives these two different regions of the kidneys this feature? The presence of, or the appearance of that granulation here is because of a component known as the glomerulus, which is where blood is filtered from. So each of these rounded structures here, within those are the glomerulus surrounded by a cup-like structure called the Bowman's capsule. Those collectively give it that granular shape. And then all the tubules pass down, dip down into the medulla and travel through the medulla, and that's what gives the medulla the striped shape appearance. Okay, so a magnified view. We move across to this side of the model where both one of the pyramids and region of the cortex has been magnified. And so this entire structure that we see here is a renal corpuscle. You see two distinct parts of it. You see that outer cup-like structure within which the blood vessels are sitting. So this collection of blood vessels is like a ball of capillaries. That is called the glomerulus. And this cup-like region is the Bowman's capsule. Together, these two make up the renal corpuscle. So look at your lab handout. What it says is renal corpuscle made up of two structures, glomerular capsule, or Bowman's capsule is its other name, and glomerulus. So this is the glomerulus, that is the Bowman's capsule. Now look at the magnified view here. What we're looking at here, this entire thing is the renal corpuscle, in which this is the glomerulus and this is the the Bowman's capsule or glomerular capsule. Now the Bowman's capsule is, can I draw in here? Okay, so the Bowman's capsule is like this. It's like this, a cup-shaped structure, into which comes the glomerulus, that is that ball of capillaries. And so for filtration to occur, blood flows in through one of these vessels, gets into the capillaries, and then it flows out. So this part here is the glomerulus, and this is the Bowman's capsule. Now, as I have drawn the Bowman's capsule, how many layers do you see in it? In the Bowman's capsule? Two. You see, this is the outer layer that I have drawn, and this is the inner layer that I have drawn. Do you see the two layers? Look at this here. How many layers do you see?
Looks like just one.
Looks like just one. And why? Because the inner layer is not that far away from the capillaries. The inner layer is sitting on the capillaries. Okay, so when you have two layers, one is outer, the other is on the organ. What do we call those two layers?
Visceral parietal.
Visceral parietal. So this outer one would be parietal. This one sitting on the capillaries. Do you see they've shown the nucleus of the cell and its processes? That's the visceral layer. That's a parietal layer. This is the visceral layer. And that visceral layer is clinging to those capillaries.
Okay.
So in the model, what they've shown is on one half over here, they've shown the visceral layer on the capillaries. On the other side, they have just shown the capillaries, just for us to understand the difference. What kind of capillaries are these that we have in the kidneys? Fenestrated. Do you see those pores that they have made on this? It's a beautifully constructed model, right? So what we've seen here, renal corpuscle, this whole thing, glomerular capsule or Bowman's capsule, this cup-shaped structure, the second inner layer of which is on the capillaries, and this is the ball of capillaries, which is the glomerulus, the parietal layer and the visceral layer. Right? Now, that's a magnified view from here. Right? From the glomerular capsule extends this tube. Now follow my pointer as I take it down. Look at this, how it's folded. Then it extends deeper down and comes up again, forming a loop. comes up again and gets folded again. These are 3 different segments of the tubule. What do we call that segment which is closer? Proximal. And because it's folded, it's called convoluted. So this part here is the proximal convoluted tubule. This is the loop of Henle. And the loop of Henle has a descending limb and an ascending limb. That leads into the distal convoluted tubule. So that's one nephron. This whole thing, tubule plus renal corpuscle together, is one nephron.
Okay.
Here is a second nephron. Right? So similarly, they've shown nephrons on this side. This straight tube, which is going straight down in the, starting from the cortex and running into the pyramid, in the medulla, is the collecting duct. And look at this here. Here's the nephron. Its distal convoluted tubule goes and opens into the collecting duct. And so this collecting duct, and that's why it's called collecting duct, is collecting urine from different nephrons. And then it takes it down through to the terminal end. And from there, urine will be poured into the minor calyx, from the minor calyx into the major calyx, from the major calyx into the renal pelvis, and out through the ureter. So filtration will occur here, right? Blood will flow in through these vessels. This is called the afferent arteriole. It breaks to form or divides to form a network of capillaries, which we said is the glomerulus. And from the glomerulus leaving out is the efferent arteriole. So blood comes in through the efferent arteriole, gets filtered out from here, and then leaves through the efferent arteriole. And that filtrate then travels through the tubule. Now, why do we need such a long tubule and so convoluted and lengthy? That's where 2 more processes are going to take place: reabsorption and secretion. Reabsorption would be any extra material which has filtered out, which the body needs to reclaim, will occur, which will be moved from the tubule to blood, and secretion will be movement in the opposite direction. You'll find that information nicely described on the module. Clearly you guys have not had the time to look at that, but when you do that. So that's the reason why the nephron is so long. And by the time the filtrate travels to the end here, all the necessary changes are made. Just to give you guys an idea as to how significant absorption is, on an average, every day, the amount of filtrate which is formed in our kidneys is 180 liters. And on an average, how much of urine do we excrete in a day? Just make a guess.
100 milliliters an hour, right?
So it's about 1.5 liters on an average per day. 180 liters is filtered, only 1.5 liters is excreted. So where does that extra water go? It's reabsorbed. In addition, whatever else the body needs, it's reabsorbed. Right? Okay, so that's a nephron. Here's the collecting duct, and distal collecting efferent arterioles. Juxtaglomerular apparatus now is this region here. So this is the distal convoluted, look at this part here, see the distal convoluted tubule? See how it's coming back close to the glomerulus and the efferent arteriole? So these two have a close association. That's what we see here. So these are specialized cells called the macula densa cells. And in the arteriole, there are specialized cells called the granular cells or juxtaglomerular cells. These two work together to control several physiological functions, one of them being blood pressure. So when you get to physiology, they'll mention this. Do you guys remember macula densa cells and juxtaglomerular cells? And you will have to say yes. If not, just tell them.
Especially if you're the teacher.
Absolutely. Okay, that's good enough for today. Go through these, and next time we'll start from here on Monday and go through the blood supply as well as do reproductive.